here, and we today are going to read an article about how life beneath the ocean surface, actually on the ocean floor, can give us some ideas about life on Mars. All right? So, the title of this article, it's from Science Daily, is Discovery of Life in Solid Rock Beneath the Sea May Inspire New Search for Life on Mars. Bacteria live in tiny clay-filled cracks in solid rocks, millions of years old. So what we're going to do is we're going to read this article, and at the end, you're going to go to the bottom of the link or to the description in this video. There's going to be a link where you can answer some follow-up questions about the article so that I can see you're participating and engaging and learning from what we're doing here. All right? So I'm going to do read aloud. I've also linked the article in the description below so you can follow along on your own, or you can follow along on the screen as I read this. So let's get started. Newly discovered single-celled creatures living deep beneath the seafloor have given researchers clues about how they might find life on Mars. These bacteria were discovered living in tiny cracks inside volcanic rocks after researchers persisted over a decade of trial and error to find new ways to examine the rocks. Researchers estimate that the rock cracks are home to a community of bacteria as dense as the human gut or stomach, about 10 billion bacterial cells per cubic centimeter. In contrast, the average density of bacteria living in mud sediment on the seafloor is estimated to be 100 cells per cubic centimeter. I am now almost over expecting that I can find life on Mars. If not, it must be that life relies on some other processes that Mars does not have, like plate tectonics, said Associate Professor Yohei Suzuki from the University of Tokyo, referring to the movement of landmass around Earth, most notable for causing earthquakes. Suzuki is the first author of the research paper announcing the discovery, published in Communications Biology. Magic in clay minerals. I thought it was a dream, seeing such rich microbial life in rocks, said Suzuki, recalling the first time he saw bacteria inside the undersea rock samples. Undersea volcanoes spew out lava approximately 1,200 degrees Celsius or 2,200 degrees Fahrenheit, which eventually cracks as it cools down and becomes rock. The cracks are narrow, often less than 1 millimeter or 0.04 inches across. Over millions of years, those cracks fill up with clay minerals, the same clay used to make pottery. Somehow, bacteria find their way into the cracks and multiply. These cracks are a very friendly place to live for life. Clay minerals are like a magic material on Earth. If you can find clay minerals, you almost always can find microbes living in them, explained Suzuki. The microbes identified in the cracks are aerobic bacteria, meaning they use a process similar to how human cells make energy, relying on oxygen and organic nutrients. Honestly, it was a very unexpected discovery. I was very lucky because I almost gave up, Suzuki said. Crews for deep ocean samples. Suzuki and his colleagues discovered the bacteria in rock samples that he helped collect in late 2010 during the Integrated Ocean Drilling Program, IODP. IODP Expedition 329 took a team of researchers from the tropical island of Tahiti to the middle of the Pacific Ocean to Auckland, New Zealand. The research ship anchored above 3 locations along the route across the South Pacific Gyre and used a metal tube 5.7 kilometers long to reach the ocean floor. Then a drill cut down 125 meters below the seafloor and pulled out core samples, each about 6.2 centimeters across. The first 75 meters beneath the seafloor were mud sediment, and then research collected another 40 meters of solid rock. Depending on the location, the rock samples were estimated to be 3.5, 33.5, or 104 million years old. The collection sites were not near any hydrothermal vents or sub-seafloor water channels, so researchers are confident that the bacteria arrived in the cracks independently rather than being forced by a current. The rock core samples were also sterilized to prevent surface contamination using an artificial seawater wash, a quick burn, a process that Suzuki compares to making buri, flame-seared sushi. At that time, the standard way to find bacteria in rock samples was to chip away at the outer layer of the rock, then grind to the center of the rock into a powder and count the cells out of the crushed rock. I was making loud noises with my hammer and chisel, breaking open rocks while everyone else was working quietly with their mud, he recalled. How to slice rock. Over the years, continuing to hope that bacteria might be present, but unable to find any, Suzuki decided he needed a new way to look specifically at the cracks running through the rocks. He found inspiration in the way pathologists prepared ultra-thin slices of body tissue samples to diagnose disease. Suzuki decided to coat the rocks in a special epoxy to support the natural shape so that they wouldn't crumble. Then, when he sliced the thin layers, these thin sheets of solid rock were then washed with dye that stains DNA and placed under a microscope. The bacteria appeared as glowing green spheres tightly packed into tunnels that glow orange, surrounded by black rock. The orange glow comes from clay mineral deposits, the magic material giving bacteria an attractive place to live. While genome DNA analysis identified the different species of bacteria that lived in the cracks, or whole genome DNA, samples from different locations had similar but not identical species of bacteria, rocks at different locations, at different ages, which may affect what minerals may have accumulated and therefore what bacteria are most common in the cracks. Suzuki and his colleagues speculate that the clay mineral-filled cracks concentrate the nutrients that the bacteria use as fuel. This might explain why the density of bacteria in the rock cracks is 8 orders of magnitude greater than the density of bacteria living freely in the mud sediment where seawater dilutes the nutrients. From the ocean floor to Mars. So how does this connect to Mars, this discovering the ocean floor? The clay minerals filling cracks in deep ocean rocks are likely similar to the minerals that may be found now on the surface of Mars. Minerals like a fingerprint for what conditions were present when the clay formed. Neutral to slightly alkaline levels, low temperature, moderate salinity, iron-rich environment, basalt rock are all conditions that are shared between the deep ocean and the surface of Mars, said Suzuki. Suzuki's research team is beginning a collaboration with NASA's Johnson Space Center to design a plan to examine rocks collected from the Martian surface by rovers, including keeping the samples locked in a titanium tube and using a CT, computed tomography scanner, a type of 3D X-ray, to look for life inside clay mineral cracks. This discovery of life, where no one expected it, in solid rock below the seafloor, may be changing the game for the search for life in space, said Suzuki. So that completes our article. I think it's very interesting, the fact that we can now slice rocks very thin and examine those bacteria under a microscope, and then maybe eventually doing that on Mars would be incredible, because then we could see the potential for life there. That would be awesome. So I hope you enjoyed this article. Next step, I want you to click the link below and take the quiz that has just some follow-up questions, so that we can internalize what we've read more. Hope you're doing well. Hope you're staying safe. I really miss you guys. Love you guys. And yeah, just keep learning. This is a very important time when just lots of students aren't going to want to learn, but I know you guys are motivated, and I want you to learn as much as you can during this time. So stay safe, work hard. Love you guys.